Hi, thank you for the opportunity to talk with you about this increasingly important topic, and thank you to the organizers for this invitation. Today, I want to talk to you about using real-world data for research and clinical assertions. So before I get started, I think it's important to understand the 21st Century Cures Act that was passed by Congress in 2016. The 21st Century Cures Act will streamline drug and device approval through modifications to the discovery, development, and delivery components of the regulatory process. Importantly, the 21st Century Cures Act has paved a pathway to allow real-world evidence outside of the randomized controlled trial to be used for clinical assertions. Furthermore, regulators must incorporate information on the patient experience. So, what does that mean? I want to talk a little bit about the difference between real-world data and real-world evidence. Well, real-world clinical assertions are generated from evidence developed from real-world data. Real-world data is data related to the patient's health and health care. It includes information on their general health status, well-being, etc. And importantly, this information is gathered from routine clinical care. This can be EHRs, registries, wearables, claims data, etc. And these data are likely to better reflect the general population seeking treatment rather than the select patients enrolled in a clinical trial. And then real-world evidence is obtained from these real-world data. These often include information on potential risks or benefits from a product, and importantly, we can assess information at the population level. So the second aspect of the 21st Century Cures Act was using patient experience data, also known as patient-reported outcomes. These are typically side effects or symptoms that can be only reported by the patient, such as headache, fatigue, depression, etc. And these endpoints allow us to incorporate quality of life and patient goals into treatment decisions. However, meaningful patient experience data is often not routinely captured in our healthcare records. Often, PROs are collected through prospective qualitative studies that are timely and expensive. When they are captured in the EHRs, they're currently captured generally in a clinical note, and it's difficult to extract this information for secondary use. This, of course, requires advanced informatics to mine the data, and therefore there's a lack of population-level data on meaningful patient experiences that can guide treatment and policy. So in my work, we use electronic healthcare records to really advance the use of real-world data. First, as you know, there's been a widespread adoption of EHRs, making it accessible to most healthcare centers. EHRs include granular data on care processes and outcomes, clinical decisions, and sometimes patient surveys, which therefore might have the opportunity to allow you to assess patient-reported outcomes. And we can use these data generally to improve healthcare delivery by developing clinical decision support tools, performing predictive analytics, or streamlining evidence generation. However, EHRs also have many challenges when we try and use them. Importantly, EHRs were developed for billing and not for research. Therefore, they're not organized by patient and episode. The reliability of documentation is variable. We have to deal with missing data, incorrect data, and often out-of-date data. And finally, the interoperability or exchange of information across systems is limited due to technical and algorithmic challenges as well as incentive disconnects. And so when the FDA thinks about using real-world evidence for clinical assertions, they take into account these challenges when using EHRs. And so they always ask, what questions can be asked using real-world data? How many patients can be assessed? And is there an opportunity to look at rare outcomes? How is inherent variations in care addressed in real-world data? How you deal with data quality, as I mentioned, the accuracy, missingness, relative endpoints. Is there a role for inferiority designs within the real-world data? And what is the pre-specified design for analysis? So let me start with one of the studies we have done using real-world data in cardiovascular medicine. Here we have developed a framework for identifying cardiovascular features in EHRs. And what I mean by that is we've developed technology to extract information from EHRs, both in the structured and unstructured components of the EHR data. And then we compare precision and recall amongst these different components of the EHR to identify what can be used for clinical assertions and what might need other sources of data. Now, in order to do this study, we got a consensus list of cardiovascular features important for inclusion and exclusion criteria in clinical trials, including diseases, procedures, medications, etc. And we used an EHR data set of about 11,000 patient records between 2010 and 2016. And so for this study, we were trying to assess regulatory grade and how we can meet regulatory grade standards using EHRs. So first we had to define what was the cutoff for regulatory grade in this study. And to do this, we reviewed several RCTs in cardiovascular medicine and looked at assertions comparing study arms. And we found that in general, we seek about a 10 to 20% benefit between arms. Therefore, we set a recall threshold of 85%. Recall or sensitivity tells me that of the total features we identify, how many do we miss? So a 10 to 20% inaccuracy and the ability to identify features roughly translates to what is being used in the randomized control arms to compare trials. We want to be sure that we aren't more than 20% inaccurate so we can meet that threshold of comparing arms. We then set the precision threshold to 90%. Now, precision tells us if of the features we identified, how many are correct. And usually we're less tolerant with precision, therefore we set a higher threshold because this could lead to inaccurate information being added to the data set. So for example, if I say somebody's taking a medication and they aren't, I'm adding information to the data set that's false. So based on these thresholds, we developed AI technologies, mainly NLP and machine learning, to identify and extract features from structured and unstructured data in the EHRs. And so what we found, this is a table of the diseases and procedures we identified. I don't want you to look at all of this information, but I think it's important to identify the overall theme that we found for this. So first we looked at diseases and procedures, and when you look at identifying hyperlipidemia, coronary artery disease, etc., we saw consistently that trying to capture this information in EHR structured data gave us very low recall but adequate precision. When we then tried to extract these features from unstructured notes, we got much higher recall, above 90%, and very high precision also. When we looked at procedures, the structured data gave us very high precision. And that's likely because these are billable procedures, and therefore they're going to be very well documented in structured data. However, for recall, we found that it was extremely low using the structured data. And then in the unstructured data, we had very high precision and recall. We also looked at medications and laboratory values. And so not surprisingly, we found looking at medications, unstructured data had very high recall and precision. These are often recorded as structured data and will be well captured in this structured data as we show. But we were also able to very accurately extract this information from unstructured data using clinical notes and text for lab results. Unfortunately, we did not have the lab results in the structured data of this EHR data set. But when we look at the unstructured data, we again had very high both recall and precision. So when defining regulatory grade, we can use real-world evidence, and this work really highlights how we can best identify different features needed to identify cardiovascular cohorts in EHR structured and unstructured data. We set measurable criteria for both recall and precision. It's important to note that the reason why recall is so often not reported in the literature is because it's very labor-intensive to capture. For example, in a study evaluating cholesterol-lowering drugs for heart attack, A data scientist may poll all patients who meet this criteria using structured data, medication lists, or problem lists. This may result in the identification of 300 patients of a cohort of 10,000 patients. Now, the data scientist would need to next review the charts for those 300 patients to confirm that the correct conditions are referred in the clinical narrative. So this narrative this assesses precision, and we can go through the 300 notes. However, to assess recall, we would need to go through the 10,000 patients and identify how many patients we missed. So you can see it's much harder to get at recall, and it's much more labor intensive. Um, so next, I want to show you another example of how we use real-world evidence to look at representativeness of randomized controlled trials. And for this one, I'm going to use an example of prostate cancer, but you can imagine you could do this for any clinical trial. So what we did for this study is there was a dream challenge, which was a crowdsourcing challenge where a group of scientists put together 4 phase 3 randomized controlled trials, and they put it out to the public, and the challenge was is who could identify the best prognostic model based on these 4 RCTs. And so the winner of the challenge identified a penalized Cox regression model that was able to predict mortality with with an adequate accuracy of 0.79. So importantly, these data were made available to the public. So we were very interested in looking at these randomized controlled trials and seeing how can we incorporate these models into our data set. So first, we looked at the demographics, and not surprisingly, we found very important differences in the patient demographics between patients actually seeking care in the real-world setting, so in our EHRs, versus the patients that were included in the randomized controlled trial. We saw stark differences across age, race, ethnicity, Carlson Index, and PSA. Importantly, if you look at race in the RCTs, they were about 90% non-Hispanic white compared to 68% in our EHRs. And they only had 1% of Hispanic Latinos in the RCT, as where we had about over 8%. So what we did is we took the model from this challenge, and they had over 101 variables included in their model. So we tried to apply that model to our EHRs, and we had very low accuracy, about 0.694. And what one of the main issues was, in addition to the demographics, is many of the variables they used were not collected in routine care. So these were like lesions of the liver, et cetera. And so we were able to use a feature optimization criteria to really reduce the number of variables that contributed to the prognosis of the patient. And so by using this strategy, we were able to decrease the 101 variables down to about 30 to get the optimal area under the curve of the prognostic model. So we then took this cohort from our EHRs, and we wanted to look at patient experience. So we were able to find in our EHRs that these patients actually completed some treatment surveys, some patient-reported outcome surveys known as the PROMISE survey. And so we had patients, these were patients seeking chemotherapy, and so we were able to find surveys looking at pre-treatment, during treatment, and post-treatment, and compare the patient experience. What we found, interestingly, was overall most patients did not change much between the pre and post-treatment. But during treatment, we saw very different trajectories of the patient patient-reported outcomes. Now, the first graph shows patient physical health, general health status, etc. And the second graph shows general mental health, depression, anxiety, etc. So by pulling in patient-reported outcomes, we were really able to identify different reactions to chemotherapy among these patients, which really helps identify information on the patient experience during treatment, which is often overlooked in most studies. So lessons learned here. randomized controlled trials often perform poorly in our EHRs, and we found that there were stark differences in demographics and also in the features used in the model. However, we were able to perform local model optimization to provide good discriminatory values. And it's important to think about the cost of features that are included in this model. And our feature reduction strategies were really beneficial to applying these models developed in the randomized control trial to our EHRs. We were able to include the patient experience data to better understand best treatment strategies. And this allows us also to assess bias and hence algorithmic fairness when we're thinking about applying these very narrow randomized clinical trials to a broader population. So in conclusion, we have developed a framework to use real-world data to generate real-world evidence for clinical assertions. When we pull our data together, we try and capture as much data, so we're not looking just at the structured data in the EHRs, but we also include the unstructured data, patient surveys, etc. We're also using advanced technologies, so we use natural language processing, machine learning, deep learning, etc., so that we can really leverage all the diverse information that's captured in the EHRs. We can then quickly and accurately identify inclusion and exclusion criteria for clinical trials, which allows us to perform observational studies, pragmatic studies, and also to start thinking about historical and synthetic control arms for clinical trials. We're setting standards for using real-world data for clinical assertions. The cutoffs that I presented to you today are really open for discussion, but we're the first ones to really set out that there really needs to be a cutoff for how we're going to say that this is a good metric for clinical assertions. And so in the end, we use real-world data to guide discovery, development, and delivery of therapeutics, and it really helps augment the gold standard of the clinical trial. And so with that, I would like to thank my team. We have a very diverse team, including clinical investigators, methodological staff, postdoctoral fellows, medical students, graduate students, etc. And with that, I will leave you with my email and our website in case you have any additional questions you would like to ask. Thank you.